Welcome, everyone. I see we've got a good audience already, and people will continue to fill in. As you know, my name is Kevin Curran. I host the guest speaker series for Caring Cross. Caring Cross is committed to bringing cell and gene therapies globally and improving patient access. We are honored today to hear from Dr. David A. Williams. David Williams is the Leland Fikes Professor of Pediatrics at Harvard Medical School and the Chief of Hematology Oncology at Boston Children's Hospital. So David's been involved with hematopoietic stem cells since the 1980s. David's had a long career. I'll just touch on a few items. He was involved in early trials with gamma retroviruses, starting to explore what's possible in terms of genetic engineering of HSCs. David was involved in cloning interleukin-11, which led to a medicine for thrombocytopenia. But most recently, and the work David's going to talk about today, is it's a novel approach to sickle cell using a lentiviral vector, and in this case, using a short hairpin RNA embedded in a microRNA. And I'm going to let David give details over that. David and I were both just out in New Orleans. There was a lot going on there at ASGCT, and we are fortunate that David is back to his office and took some time out of his day to speak with us. So, David, I will hand it over to you.
Thank you. Thanks so much. And I'm very pleased actually to be able to share with you some of our work. I'm very excited about the work and excited to present it to folks at Caring Cross with the goal in the future of expanding access, particularly potentially in places other than the United States. Let me see if I can move forward. Here we go. Here's a disclosure. I'll just make one that's related to what I'm going to talk about. You'll see at the top bullet, Bluebird Bio actually provided the GMP formulation of the vector I'm going to talk about as part of a previous license agreement, but they've had nothing to do outside of that with the trials I'm going to present. So I'm going to just give you a very, very, very brief background on sickle cell disease gene therapy. I'm going to talk to you a little bit about how we engineered the shRNA to target BCL11A, the concept. We did a pilot and feasibility study and did some additional biological studies I'll touch on, moved into pivotal phase 2 multi-site trial. And then I'll end with a couple of thoughts about new approaches to improving in manufacturing and improving the vector even further. So all these are relatively short sort of subject vignettes, and so I'll try to get finished in about a half hour. So as many of you know, there are now multiple genetic approaches to treating sickle cell disease. Gene addition, which is the Bluebird Bio approach, where lentivirus is carrying a new globin gene that's been approved by the FDA. You're aware of gene editing, CRISPR, approved by the FDA's Casgevy. We're a site for Beam. We're doing base editing now in sickle cell disease. It's working well. And then I'll talk to you about the shmiR technology, as I mentioned. Now, in spite of the fact that we have 2 FDA-approved products, there are some challenges that are remaining with respect to taking advantage of those and implementing those. So one you're aware of is that there was probably development of leukemia in the early Bluebird trial, and it's raised the question still unanswered as to whether patients with sickle cell disease have a cancer predisposition, and that held up our trial for almost a year, and is still being answered in several ways. I'll just touch on that briefly later on. One big roadblock that we found in the commercial center products we are a center for both Bluebird and for CRISPR, Vertex, is obtaining enough hematopoietic stem cells for manufacturing and the timeliness of doing that. And then, of course, you're probably aware that the cost is really quite dramatic. And so having access is an issue. So I'll touch on these during the talk. I'll talk now about the shmiR technology. But the first thing to talk about, just very briefly, is why is there a lot of focus on fetal hemoglobin as a treatment for sickle cell disease? And the first thought about this actually is quite old. So you can see this publication in the AFCR in 1948 from a pediatrician in Brooklyn named Janet Watson. And she noticed in the newborn nursery that the babies of patients with sickle cell disease had strikingly lower numbers of irreversibly sickled cells on their blood smears than the mothers. And so she hypothesized in this paper that that may have something to do, as you can see at the title, with fetal hemoglobin. And this is then the physiology of that situation. So you can see to the left of the dotted vertical line is in utero, to the right is postnatally. And in utero, we all express alpha globin, and we express as the beta-like globin gamma globin shown here. Let me just get my laser pointer. So here's gamma globin expression. And then after we are born, miraculously, gamma globin expression is shut off and beta globin expression turns on. And when you have sickle cell disease, that beta globin expression is the mutant sickle beta globin. And so before this switch, an infant has no symptoms at all. When I see these patients in my hematology clinic, I can't tell that they have sickle cell disease by their phenotype. Then after that happens, then they become symptomatic. Let's see. So for many, many years, there was a lot of work to try to understand the regulation of fetal hemoglobin. And the NIH spent millions and millions of dollars studying the locus, and that was fruitful. But the real finding that turned kind of the field around is shown on the left here, which was GWAS studies, in which surprisingly, this is a Manhattan plot. So to read this, you look at the vertical lines, dots here, and you see that there was an association between genomic variations in this gene called BCL11A and fetal hemoglobin levels worldwide. So BCL11A is a transcription factor. It's essential for normal B cell development. But before this study, there was absolutely no indication or thought that it had anything to do with globin expression. And so from a lot of studies cited here on the right, it became clear that BCL11A is a master regulator of this physiologic switch. And so after birth, you have high levels of BCL11A that retards the expression or inhibits the expression of gamma globin. So you have lots of adult hemoglobin alpha 2, beta 2. So our thought was to use this physiologic switch as a therapeutic target so that you see, once again, represented here in utero, you have these enhancer units of the globin locus called LCRs. They're interacting with the gamma locus here. There's two gamma genes, G gamma and A gamma, producing fetal hemoglobin. And then after birth, you turn on, these enhancers interact with the adult beta globin locus, in this case, sickle, so you turn on sickle cell disease. So the therapeutic target then would be to reverse this switch and turn fetal hemoglobin back on. And the idea was to use shRNA or RNAi in a lentivirus vector to induce gamma globin expression and silence simultaneously the beta gene. And the advantage of that is that the anti-sickling fetal hemoglobin is increased while the defective sickling beta globin is downregulated, providing actually synergistic effects. In other words, you're getting two for the price of one. And because this is a cellular physiologic switch, the ratio of alpha to beta globin chains remain balanced. And that's quite important because otherwise you run the risk of inducing a thalassemia-like syndrome. So we developed a lentivirus vector we call the shmiR vector. And this is a standard lentivirus for those of you not in the gene therapy field. So the lentivirus has it's an HIV-based virus. All the HIV sequences essentially are removed. And the expression of this shmiR is driven by the beta globin promoter itself. And the targeting shRNA in red is embedded in a microRNA scaffold. That's what we call the shmiR. And that sort of schematically, this is what it looks like. So the microRNA is in red and the shRNA is in blue. And the advantage of this is it mimics endogenous microRNAs in the sense of how the cell processes it, which leads to more physiologic levels that reduces non-specific shRNA toxicities. But more importantly, now it allows us to regulate the expression specifically in erythroid cells. So this avoids the toxicity that we knew we would have in B cells because it's a transcription factor in B cells. But in our studies, we found that also BCL11A is required for appropriate stem cell engraftment. And so that's the other toxicity, that on-target toxicity that we would have. So this allows now, the way we engineered this, erythroid-specific expression, so only expression in erythroid progenitor precursors and red cells using this vector. So this led to phase 1 first-in-human study, where we call this a post-transcriptional silencing of BCL11A. So it's not editing the genome at the BCL11A locus, it's actually taking advantage of knocking down the RNA. Excuse me. So this was first in human study. There was originally 7 patients intended to be treated. It went so well that we increased it to 10 patients in different age cohorts. The inclusion criteria are pretty standard for an actually allo transplant in these patients. So lack of an HLA sibling donor, a severe phenotype, failure of hydroxyurea. And originally we included patients with a history of stroke, but we subsequently modified that inclusion criteria, exclusion criteria. And this was funded by the FDA. And as I mentioned, the GMP vector, although it was our vector, it was the product was actually made by Bluebird. So this is the protocol. It's again pretty standard. We mobilize the stem cells into the peripheral blood using the mobilizing agent called pliclixofor, as described here in this manuscript. And the CD34 cells are selected over a clinically grade column. They are taken to a GMP facility where the cells are transduced. The release criteria are done, and then after that's completed, patient is admitted for conditioning with full dose, essentially full dose busulfan. This sort of schematized that. So here's CD34 enrichment. The target for us is really 10 million CD34 cells per kilo. Then the cells are put into a closed system for pre-stimulation, transduction, and cryopreservation. And then our release criteria include a minimum of 4 million CD34 cells per kilo. I'll talk a little bit more in a minute about the product release criteria. So the results of this, initial results were already reported in New England Journal. It's called post-transcriptional genetic silencing of BCL11A. And this was really the first, excuse me, validation of BCL11A as a therapeutic target in humans. Now, one point is this was a phase 1 toxicity study, so we paid a lot of attention to adverse events. And basically, this is a very safe product to this point. So there were pre-transfusion grade 3 AEs that were related to placement of the CVL and mainly thrombosis related to lines. There were no grade 3+ adverse events related to the mobilization or collection itself. And there's been no adverse events in the follow-up due to the medicinal product, that is the gene therapy product itself. And the longest patient in this initial study is now well over 5 years. And we've watched very, very carefully for any signs of insertional abnormalities. And we've seen none to the date, following these patients very closely. I do want to make one comment to contrast this approach with editing. And this is the first 10 patients on the study. And it has to do with the challenge of obtaining enough hematopoietic stem cells for manufacturing timeliness. And so in this trial of the first 10 patients, we collected enough stem cells in one mobilization cycle in 9 out of 10 patients, and the other patient required 2 mobilization cycles. And you can see that in all instances, save one, we attained our product minimum of 4 million CD34 cells per kilo. So this is really an excellent success in collection and manufacturing. And if we actually look at the efficiency overall of the process, this is the data I just showed you. And then if we plot the amount of cells collected versus the amount of cells we get at the end, you can see that the product efficiency is over 60%. And again, this is quite good compared to the product efficiency that we've been seeing in the commercialized products. And to kind of emphasize that a little bit more, this is the famous trial on one patient in editing, or the first patients done with CRISPR editing. And if you look at the table in detail, you can see that in both the full analysis group and the primary efficacy group, that the number of mobilization cycles required varied from 1 to 6 in the full analysis group and 1 to 5 in the efficacy group. And this has been our experience here too with this product, which is you have to collect lots and lots of cells. And that requires, each of these cycles requires a minimum of around 4 weeks in between. So you can see if you have 6 cycles, that's 6 times 6 months of waiting to get the product to go into manufacturing. So half a year just to get enough cells to start the manufacturing process. So we have patients that have been waiting quite a long time to get their product. So this is sort of a distinction of using lentiviruses, where the toxicity of the process is much less than the toxicity of electroporation. I mentioned already the release and acceptance criteria. Because this is the second trial is a pivotal trial, the FDA looks at this quite carefully. So you can see here the things that we do to determine the product is ready to go. Cell count is obvious, viability is obvious. The phenotype of the cell, so CD34 cells, is obvious. The transgene is the correct size, sterility and endotoxin and mycoplasma, all obvious. The one thing that that we did, which was I think quite unusual, was to show that we were hitting our target and actually inducing hemoglobin F. For all the products, we actually do single cell analysis of hemoglobin F. And you can see we look at the number of colonies that have greater than 50% hemoglobin F. And a release criteria is actually the number of colonies in which the hemoglobin F is over 30% of the total hemoglobin. That's the amount that's hypothesized to be required to inhibit sickling in the deoxygenated state. In the phase 2 trial, we also, at the FDA's request, added an in vitro sickling assay as part of the release criteria. And this is kind of problematic because there's no validated in vitro assay for sickling, but we're doing that and collecting the data. The amount of vector in the cells and the fact that there's no wild type lentivirus, all these are what are release criteria have to be fulfilled before release the data. So here's the informative data on those 10 patients that are in the phase 1 trial. Here's this 30% cutoff in kind of blue dashed lines. And you can see amazingly that the very high number of erythroid colonies that have very high induction of fetal hemoglobin using this, saying that the vector very nicely targets and induces fetal hemoglobin. Now, one thing that we learned from this dose-finding phase 1 study is you have to have a certain amount of vector in the cells to induce enough fetal hemoglobin in enough cells to be phenotypically corrective. And so we had in these 10 patients, one patient that we deemed of treatment failure. And you can see that patient has the number of vector copies in the cells, known as VCN, is low. It's about 0.2. And so this really helped us for the phase 2 subsequent trial. We modified our manufacturing processes to ensure, as best as we could, that there would be no patient who would have a vector copy number below 0.5. One additional point I would make is that in contrast to the Bluebird product, where you need multiple copies of the transgene to induce or to have maximal effects, in this vector, it's so potent that an average VCN of 1 maximally induces fetal hemoglobin. And so that is an added safety feature, because the more insertions you have per cell, theoretically, the more risk you have of insertional mutagenesis. So we utilize these pilot results to modify manufacturing with resulting uniform improvement in vivo VCN and hemoglobin induction in the ongoing phase 2 trial. And this is, as I said, is our cutoff here in red. So there were some additional things just to mention in brief. I won't go into a lot of details, but after the trial was initiated, as a result of the leukemias in the Bluebird trial, which uses a completely different lentiviral design, the funding agency, National Heart, Lung, and Blood Institute, required us to institute genomic analysis for mutations in genes associated with leukemia. Here in our institution, that's called a rapid heme panel. It's about a panel of about 80 genes implicated in clonal hematopoiesis. And because this trial was well on its way to being done, we had to go back and look at post-enrollment and post-treatment in all subjects using bank specimens and prospectively in the remaining patients. And so to date, we've analyzed now well over 60 samples, it's much more than that now, of these 90 genes. And we've seen one case in which there was a mutation retrospectively present at baseline, and then continued to be present after engraftment in one of these clonal hematopoiesis genes. That patient has done quite well, hasn't had any problems at all, is now almost 5 years post-infusion, has continued to do well. The clone itself increased a little bit after infusion, but has remained then stable since that time. And then we've looked very, very carefully at clonal insertion site analysis. That's where you look for insertion of the vector that leads to amplification of cells because of a neighboring oncogene. And we see absolutely none of that. In fact, our largest clone, if you want to call it that, is much less than 5%. I think 3% is the largest clone we've seen. So that's in contrast to instance where you have lentiviral or gamma retroviral insertion that causes leukemia, where the inserted gene amplifies the clone to 50, 60, 70% for some period of time before the leukemia evolves. So quickly to conclude, we enrolled 10 patients. We actually enrolled 11 patients. One patient dropped out before infusion. 10 patients were infused. Long-term follow-up is over 5 years. The vector works really well. It's extremely efficient transduction. There's no adverse events related to the vector. the expression. I didn't show you the data, is lineage specific, and there's stable gene marking now over all those years. The shmiR leads to broadly distributed hemoglobin F. That is, not only is hemoglobin F higher in the blood, but if you look at the number of red cells that are expressing hemoglobin F, it's high and broadly distributed. And importantly, in those cells that have F, the amount of hemoglobin F in the total hemoglobin is quite high, so over 10 picograms in most cells in most patients. And there's been significant attenuation of the sickling phenotype. We're preparing the long-term follow-up of these patients right now. So there's a couple important correlative studies that came out of this. I'll just draw to your attention in case you're interested. One is we did single-cell analysis of red cells from the patients with gene therapy, and we matched them with patients who were on hydroxyurea who had the same level of total blood hemoglobin F, and asked the questions: if you reduce oxygen in these patients, in these samples, do you see hemoglobin polymerization, and to what extent is that different? And in this paper in Nature Communications, what we show is that the patients participating in the gene therapy trial have less polymer formation at any given oxygen concentrations than patients that are on hydroxyurea. And we hypothesize in that paper why that is. And then, because it's of general interest, the whole issue about patients with sickle cell potentially having an increased risk of myeloid leukemia, we actually did single-cell DNA analysis, asking the question: does cell manufacturing, does the vector transduction, or the engraftment change the mutation burden in sickle cell patients? And what's the baseline mutation burden? That's published in this Nature Medicine paper. And what we saw in some patients, there was indeed an increase in the mutation burden. I think in 2 out of 6 patients we saw, but it was really, really low, the frequency, and there was no correlation with whether the cell had an insertion in it or not. So this requires a lot more follow-up, and so we're doing that with a Gates Foundation grant, in which we're looking at the genomic landscape in sickle cell patients compared to ethnic control populations in the UK, in Tanzania, in Uganda, and in Boston. And then we're participating in an NHLBI-funded large-scale genomic analysis led by Coleman Lindsley at the Brigham and Women's Hospital to answer this question in more detail. So all this has led to the pivotal phase 2 trial. It's called the GRASS trial, Gene Therapy to Reduce All Sickle Cell Pain. This is, again, an NHLBI and California Institute of Regenerative Medicine funded multi-site trial with myself and Mark Walters as study co-chairs. And it's called GRASS because the primary endpoint, which we negotiated with the FDA, is to determine if the single infusion of treated patients of CD34 cells treated with the shmiR vector leads to complete absence of severe VOEs in the period of 6 months to 2 years after gene therapy. So this is multi-center trial shown here. The PIs are listed. And the trial was on hold for a full year by the NIH because of this leukemia event in the Bluebird trial. The FDA never put it on hold, but the funder put it on hold, which really set us back, unfortunately. But we've now have the full accrual target of 25 patients. We've now validated a second lot that's been produced in a GMP facility that's commercially compatible. We've manufactured 24 products. 21 patients have been infused. The trial has gone extremely well. There's been no safety signals to date. But due to the requirement by the NIH, we continue to do additional genomics safety surveillance. And in this trial, in fact, if you had a mutation in any of these clonal hematopoiesis genes, you could not get into the trial. And luckily, none of the patients that we've screened of the 31 patients, none of them have had pathogenic mutation. And because of this fact that this is a pivotal trial, we've added additional safety and oversight committee. So there's an independent appointed genomics committee that adjudicates all the genomic data that we've gathered. There's an independently appointed VOE adjudication committee, that is, any pain event that is registered either before or after infusion of the cells has an independent committee of sickle cell doctors who look at the episode and determine whether it's a true VOE in their opinion or not. There's an independent appointed data safety monitoring board appointed by the NIH. There's independent clinical monitoring of both the clinical effort and the manufacturing effort. So these things are all built in so that we can be assured that if the trial hits its primary endpoint, that it will pass muster for a BLA in the future. So going forward, we've done a couple, we haven't sat still, we've done a couple more things. So I mentioned this paper in Nature Medicine, but since that time, we've now added additional changes to the manufacturing process that we've shown in this paper published last year in Blood Advances, reduces DNA damage, reduces ROS generation during the manufacturing process itself. And we're hoping in the future to include that addition into the manufacturing process. We've also developed additional vectors based on the current vector to further induce F induction and reduce the concentration of sickle hemoglobin in cells. And the nice thing about this is, because we work with Don Cohen, we've been able to generate a vector that increases the effective transduction of CD34 cells so that we can actually generate more transduced cells at less viral vector soup, which is going to reduce, of course, the manufacturing costs. And this last vector reported in Hart et al. Reduces the amount of remaining hemolysis in the sickle model mouse to the point where it's basically equivalent to a completely normal mouse. So that's quite exciting. We're excited to try to develop that further. And I won't have time to talk about that, but of course, if you think about the ability to move this into other parts of the world, in vivo delivery might be preferable. And with funding from the Gates Foundation, we're developing viral vectors that can be used to transduce hematopoietic stem cells with an in vivo injection. So that work is currently under review, scientific review. So overall, what I'd like to say is that we've introduced this new shmiR technology, which could be applied to many different genes. And in fact, in the modified vector I ended with, we can tandemly array shmiRs so that we can knock down multiple genes at the same time. And at the same time, the vector still works to put in a transgene, so you can knock down multiple genes and overexpress genes at the same time. This vector appears to be quite safe, no evidence of genotoxicity. The cell manufacturing has been remarkably consistent, with cell targets sufficiently attained in all the patients so far. The pivotal phase 2 trial is going quite well. Target accrual has been obtained. There needs to be additional study to define the genomics in this group of patients. And although 25 patients is not very many, or 35 total, it doesn't seem as if that screening has been potentially very fruitful at this point. We've introduced now multiple process improvements that further increase the efficacy, and these may be, in fact, applicable to other ex vivo approaches, other diseases. And preliminary evidence, I didn't show you this, but the potential of development of in vivo approach is certainly there. So this is, of course, a very big undertaking. We actually really appreciate the fact that many patients in the sickle cell community and the community itself has been enthusiastic and has allowed us to to accrue quite rapidly to the trial. Here in Boston, the PI of the trial has been Erica Esrick. We've had enormous help from John Manis, who is our apheresis expert. And we couldn't do this without a translational lab. This is a lab that develops and then implements assays that a standard hospital laboratory won't do. So it's really essential for the carrying out of these kinds of studies. And also our CMCF, this is a GMP lab at Dana-Farber, does all of our cell processing. And I will say that we've had enormous collaborative help from Bluebird, even though they weren't part of this trial. They've constantly been a good partner with respect to giving us advice and us giving them advice, so two-way street. So we're very appreciative of that. The phase 2 GRASS trial, the protocol officer, enormously helpful, has been Mary Epen. The data coordinating center is at the Medical College of Wisconsin. We're the clinical coordinating center, and the manufacturing facility has been here at the Dana-Farber. And the gene therapy program at Boston Children's is quite advanced. We've now done something on the order of 25 human trials in gene therapy. We're a commercial center for all the approved gene therapy products, and we have a group of leaders, a clinical research team, clinical investigators, and a translational component to our trial. We work with a lot of companies. So let me stop there. Hopefully that leaves us some time for questions if there are any, and I appreciate the opportunity to provide this short review of our data.
Yeah, absolutely. Thanks so much, David. That is quite a story, and I applaud you for pursuing this this novel approach for the sickle cell community. I forgot to mention at the beginning, for our audience, please use the chat, the Q&A, and type in a question. I can already see a question in there. I thought I would kick this off in just kind of a very broad sense. You know, you described some of the advantages, you know, when you were talking about the manufacturing, the efficiency of this lentiviral approach compared to now there's 2 commercial options, I guess, durable options, as well as standard of care. How would you see this this potential medicine, thinking in terms of like the target product profile? What would be the ideal sickle cell patient subset that would be, I guess, most appropriate for this shmiR lentiviral approach? Do you have a sense for what subset of patients would benefit the most here?
I think it's the way we think about it is sort of these 3 processes, 2 approved, 1 not, obviously, as being relatively equivalent. The advantages are the advantages are that we tend to be able to, you know, make the product much faster and have it infused much faster. I think the other thing that we're thinking about is that the commercial products, in the past at least, have not been approved for patients who've had previous CNS disease. And we've had that in our pilot study, admittedly not on our second study, but we think that's still an area that needs to be further explored.
Yeah.
And I think the virus itself is excellent, and the transduction efficiency is excellent. So I don't know that on an individual patient you can say one versus the other, but I think globally it provides an attractive alternative. And I think, you know, we believe that the manufacturing costs for this would be substantially less than the manufacturing or the cost of the current approaches. But that's something that we have to work on some more.
Is that just kind of incremental efficiencies that have been built into lentiviral vector manufacturing, or what else it allows for that, I guess, cheaper cost of goods?
Yeah, I think one is the titer the vector and how much vector we have to use. And the other is that each time you have a mobilization cycle, that's it's potentially an emission, it's a central line placement, it's all that and a mobilization cost. So all that adds up to the cost of the final product.
That makes sense. All right, let's. The first question here was from Supercharger Moses, he's a big fan of the Caring Cross community. He's would like you to throw a little more light on the one treatment failure due to a low in vivo vector copy number. Can you elaborate on that?
Yeah. So the way the shmiR works is that for it to work effectively, you have to, in many cells, you have to knock down the target, BCL11A, to below 50% of normal. And so in that patient, the cells that got the vector did that, but the problem was that the engraftment of the stem cells, the number of stem cells that had the vector in it was insufficient to have broadly the red cell population have high F. And it turns out that that patient, this happens, I've been doing gene therapy for a long time. From time to time, you see a patient where the manufactured product is good, but the engrafted product is suboptimal. That was the case in this patient. Now that patient is better. It's not as if the patient isn't better, but the patient continued to have pain episodes after the infusion of the cells, which we considered to be by protocol a failure. Since we've changed our manufacturing, we haven't had in the 20 some patients that have been infused so far in the phase 2, we haven't had anybody had low VCNs in vivo. So while we don't quite understand on that one patient why the in vivo VCN wasn't good, we upped the dosing so that we are not having that on any patient so far as an issue.
Okay, yeah, thanks for that. And then the second part of Moses' question, it really goes to us at Caring Cross, where they're saying, how can Caring Cross help unpack these hard science sessions? You're for global community advocates and patient leaders. So there's, as I mentioned, there's some percent of our audience that are patient leaders and involved closely with sickle cell globally. So some of this is homework for me to get into. I mean, I've talked a lot about this group to lentiviral vectors, but the shmiR piece it's an additional piece of biology that I think people in this, like the sickle cell patient community, are probably mostly unfamiliar with, because you need to start thinking in terms of viral vector insertion to chromosomes, and then also this sort of regulated RNA piece. And I think maybe you can help me unpack this. If we were to think about the cell biology of this, you've got the short hairpin RNAs being expressed, and now they're gonna. Maybe you can describe how they're going to be engaging and maybe the persistence, because I was curious, how long will they hang around and be able to be active in the cell? Can you speak to that?
It's great questions. Yeah, it's great questions. Yeah. So take the latter one first. It is an integrating vector, so it's permanently part of the genome, and it'll continue to express, as far as we know, for decades to come. And we have no indication at all. We're carefully monitoring that there's any decrement in that as time goes on.
Because you have that red blood cell specific expressor, that's going to drive the expression of the shmiR in the red blood cell.
Only in the red blood cells. So the stem cells got the vector, but it doesn't turn on until red cell differentiation begins. And the way it works is that the cell normally processes these things called microRNAs. They're just that, they're short RNAs. And That's part of the normal cellular phenotype biology. And what we're doing is we're basically targeting the messenger RNA for the target, which is BCL11A, so that when that shRNA is made, it binds the mRNA and that's degraded. And the reason that we had to put it in a microRNA is so we could regulate it only to be expressed in red cells and not other cells. So the biology, the trick in the engineering was to sort of think of ourselves, if we don't try to express it as an shRNA, but we express it as a microRNA, then the cells will take that and appropriately process it. And it should allow us to then hook it up to a promoter and enhancer that only expresses in red cells. And this really eliminates the toxicity that you would see if it was expressed in, for instance, in B lymphocytes, where you need BCL11A to make a B cell, basically.
Okay. Yeah, that's helpful. And also I'm going to address this further in the upcoming year, as this technology kind of advances and as we follow along with your phase 2. We'll keep talking about this different approach to lentiviral. I want to keep hitting these other questions here. From Leon Shalolo, the question is, have you had an experience with this new approach in patients living in an environment where malaria and other infections are constantly present?
Oh, that's a really good question. I assume what he's thinking about is whether F has an effect on malarial infectivity in the red cell. No, we haven't had any. I think it is theoretically very interesting from a biology standpoint, but we haven't had that experience yet. This is all 35 patients are all in the United States at this point. Yep.
Sure. Okay. And then Kartik Chetlapalli had the same, similar clarifying question. For the phase 2, will the cell therapy product be manufactured centrally or at sites associated at GMP? So is this a place of care or a centralized approach?
Great question. We started out having 2 sites here at Children's, Dana-Farber, Boston, and UCLA. UCLA manufactured one product, and then we've manufactured the other products. But having said that, what I'm quite interested in, and hoping we can do this with Caring Cross, is to convert this with our know-how into point-of-care manufacturing. And I think, personally, that's a way to develop a less expensive product and adapt this to other places where we will need to, work together to make sure the manufacturing is primo, is the best it can be, but then have it manufactured at the location. Obviously, this won't be every location, but it can be lots of locations rather than having central manufacturing. Central manufacturing is the way drug companies do this, but it is a bottleneck for sure. And I think if you think about not just sickle cell disease, but many of the other rare diseases that can be approached now with genetic therapies, there's going to be many of these that companies won't have anything to do with. And so we're going to need to develop, and I'm really invested in developing point of care delivery. You guys have already had experience of that with CAR T cells. And I think that, for instance, the people in Spain have shown that that works really quite well and reduces costs significantly. So that's sort of my dream for this, if we can pull that off. And I'm not sure, the companies that are making the commercialized products, I'm not sure that I don't know for sure, but I'd be surprised that they have that in their thought process right now, at least.
Right. Yeah. And that speaks to the second part of the question, which is, what is your vision to eventually out-license the therapy or point of care? It sounds like this would be a good fit where you could have the viral vector itself could be manufactured centrally, the way, we are doing that in Maryland, but that's easy to ship globally, and then you could have infusion sites and the apheresis, that place of care could be receiving the vector and then bringing that into the workflow
Yeah,
regionally.
we'd have to transfer the technical know-how about how to do transduction. And part of the technical know-how is basically how to collect stem cells from patients with sickle cell disease. It's not so easy, but all that could be packaged into a process of tech transfer. And this could be done, if it's set up appropriately, at centers around the world that have the capacity to do auto transplants. And that's kind of the vision, because I think I don't know, I think just seems unlikely to me that patients in other parts of the world are going to be able to partake in products that cost $2 to $3 million a shot. So
I would agree with that.
yeah.
Yeah. Well, it's great to hear you're thinking outside of that box. I think we have to at this point. So that opens up a lot of possibilities when you step outside of that kind of the traditional pharma box. Maybe I'll ask one last question and then we'll let you go. I saw you mentioned that you are using the busulfan as a conditioning regimen. I think we had an earlier speaker on they talked about trying to move off of that and use like a slightly less harsh conditioning regimen. What are your thoughts on that? Is the busulfan, is that going to be tough to avoid? I think our audience is probably familiar that there's some adverse events. It's low probability, but there's some issues with that as a conditioning regimen. What are your thoughts? Is it critical to keep using busulfan, or is there possibilities?
I think it's right now, it's critical that you are making sure that there's no mixed chimerism at the end of the process. I think the problems with leukemias and sickle cell outside of gene therapy have occurred in transplants that were milder conditioning, where there was basically mixed chimerism after the conditioning. So, but I think another way to answer your question is busulfan is a limiting feature of all these gene therapies. And so being able to develop a less toxic but completely myeloablative approach is preferable. And of course, the way lots of people are trying to do that is with immunoconjugates rather than with chemotherapy, where you don't have the long period of cytopenias that requires essentially ICU level admission for 30 days. So those aren't quite ready for prime time yet, but we and other people are certainly hoping and working on those. So I think there's if the thought approach is to reduce the intensity of the conditioning, I'd worry about that, because I think you don't want to end up with endogenous cells that are still there after the conditioning that are competing with the transduced cells. And in that situation, I think you worry about the damage to those cells that are still around that occurred without full conditioning. But on the other hand, everybody recognizes, if you could eliminate that altogether, you certainly could go a step towards having this done in places where you simply can't admit someone to a transplant unit for a month while they're waiting engraftment of the cells.
Right. Okay, that makes sense. And that's another good topic for us to explore with the educational talk, is thinking about busulfan and alternatives. So appreciate your thoughts there. David, this has been wonderful. I know our audience really appreciates that. I'm already hearing that from some people. So again, thank you for your time. This looks really exciting. And
thanks for inviting me. I'm really happy to have the discussion with you guys.
Yeah, and we'll be following along as the trial continues. So
okay.
we'll be in touch.
Okay, thanks.
All right, take care, David.